Chris is going to graciously kick this off. Just a brief introduction. Chris is a staff scientist in the biology R&D group at Thermo Fisher Scientific. He's been with the team for the last 14 years, starting off as an intern. His formal training is in biochemistry and biophysics, but over the years has led multiple projects developing fluorescent probes, labeling technologies, and cell-based assays for flow cytometry and imaging applications. Examples of the products that Chris has launched include live-dead fixable viability dyes, CellTrace Violet, and more recently, the Frodo AM Aster reagents and assays for antibody-dependent cellular phagocytosis applications. Chris is currently focused on improving the development of cell therapies and therapeutic antibodies. He's also a very active member of the Thermo Fisher Scientific Scientist to Scientist program, interfacing with our customers globally through webinars, presentations, and site visits when permitting. Not last year, but hopefully we'll resume that in the future. With that, I'll turn it over to you, Chris.
Great, thank you, Alexia, for the kind introduction, and really a thank you to everyone at the Knight Institute for continuing this spirit of collaboration. And it's great to get together virtually and share our science. So I've really enjoyed the first few sessions, and I really appreciate the opportunity to share some of the research our team has done really over the last many years. We'll go back into the archives here and look at the discovery of some reagents, and especially focus on not just how these different methods work, but talk about how they were discovered and how the collaboration between chemists and biologists and our business partners led to the introduction of some new technologies. And so I think everyone knows their way around Zoom by now. There's a chat function. You're welcome to ask questions as we go, but we'll definitely have time towards the end after both Bruce and I present to have more open discussion and questions. As Alexia said, I'm a staff scientist, and my interests are largely focused on developing functional reagents that help those developing immunotherapies and cell therapies to better understand how those therapies are functioning. I realize immunotherapies are very much in the popular press now. They're not so much limited to just an immunology conference, but I think just a brief introduction to what these are and how they work will be helpful. So as I understand, during Bruce's seminar, he'll discuss meiosis and how cells divide and how we can use various reagents and methods to study cell division. We can also consider the opposite, where we prevent cell division. And many chemotherapies for treating cancer, their goal is to actively prevent dividing cells from dividing. And so that when we give these to cancer patients, what we're doing is extending the lifespan of those patients. But you see, in a common plot in medical research is called a Kaplan-Meier curve, where we look at the percent of patients that survive after a given amount of time with a certain therapy. And with the advent of targeted therapy, where we're actually genetically sequencing a patient's tumor and providing an appropriate treatment, we can actually buy patients more time, essentially. But with the introduction, really in the last decade, of checkpoint blockade immunotherapy, something amazing was demonstrated, and that's that this line doesn't go all the way down to zero. We're actually beginning to use the C word, the cure word. We're curing patients now using these therapies. And it turns out when we combine multiples of these therapies, often with targeted and chemotherapies, we're actually able to, in subsets of patients, really prolong the lifespan or cure a substantial fraction of these patients. So just one hallmark study looking at nivolumab and ipilimumab, both of which contributed the discoveries of these led to the 2018 Nobel Prize in Physiology and Medicine, because as we see in patients with late stage melanoma, the combination of these two checkpoint therapies, one that binds to T cells and one that binds to cancer cells, dramatically improves the outcome for these patients. And so as I'm interacting with our customers, who are very often in this space, we try to solve challenges that they're encountering in their research. And when we look at, say, a T cell and its interactions with the generically named cancer cell, in this case, these cells essentially talk to each other. They recognize each other via the upregulation and downregulation of receptors and ligands on their cell surface. Those checkpoint blockade immunotherapies block certain receptors on either of these cells, allowing these little Pac-Mans in the T cell to target and kill the cancer cell. And so in the process of studying this interaction, for example, using common cell biology techniques like fluorescence microscopy or flow cytometry, a fundamental challenge that these researchers have is just distinguishing these cells from each other in a cell culture vessel, for example, so that we can track the behavior of both our immune cell and our cancer cell and study their interaction with each other. Now, if we combine a population of cytotoxic T cells with a leukemia cell line and just look at them under a white light microscope, they look very similar. We can see some differences, but we know this is really where the power of fluorescence-based reagents comes in, because we can actually label the two different types of cells and very readily distinguish the two from each other. There are various methods to accomplish this that have been used over many years. One, of course, is to deliver fluorescent proteins to those cells. So in this case, our T cell has been transfected with a green fluorescent protein, while our target cancer cell expresses blue fluorescent protein. And so this gives us the benefits of having very bright and long-term stable fluorescent signal in two different cell types. It also gives us something valuable, and that's the ability to kinetically track these cells over time and through time and space. However, there is, while this has been very much democratized, there's still expertise required, and there is a lot of time involved in creating these specific cell lines that perform correctly. There are other ways to track cells in different populations. Another way is just simply using fluorophore conjugated antibodies with flow cytometry. And in this case, we can use two antibodies to identify our natural killer cells and resolve those from our T cell population. And again, these are very bright and stable reagents. In this case, there's minimal expertise required to accomplish this. However, this is an endpoint experiment. We do all of our labeling and analysis at the end, so we have no idea what happened in the middle of that experiment. And also, when we're talking about immune cell-based assays, they often happen very fast. These T cells and natural killer cells kill very quickly. And so if we add on a lot of labeling steps at the end of our experiment, we may have missed something that happened early on in the experiment. And so now I'll take you back about 15 years to an experiment that was done by Bruce Lyons in Australia, where he took a dye from a small company called Molecular Probes in Eugene, Oregon, called CFSE, and he labeled spleen cells from a mouse and stimulated them. And this is the figure from his notebook where he said, after growing these cells for several days, do these separate peaks represent cell divisions? And he called out this intriguing pattern of fluorescence intensity decrease. What he actually discovered at that point is what we now call cell proliferation analysis by dye dilution. And what he discovered is that if you take a tracking dye and label a population of cells, so they're very bright, in this case with flow cytometry, every time those cells divide, half of the dye is passed to each daughter cell. And so they become half as bright. One of the early reagents I was involved with at that point in Vitrogen was a reagent called cell trace violet cell proliferation kit. And here we can see we're able to track cells, in this case, over 8 days of them growing in culture, and see very clearly that they've passed through several different generations. At the time, we had thought this would be useful, and I'll mention this is synthesized by a very great chemist, Zhizhang Liu. At the time, we thought this would be useful for this experiment, and that's tracking the proliferation of cells. Later, we realized that this class of dye is very good at tracking cells through space and time, as we see in this wound healing assay done by Kevin Chambers, using a dye developed by Wenjun and Mike. Now, these cell tracking dyes solve all 4 of our criteria. They're bright and stable. We can analyze them over time for kinetic analysis. Very little expertise, just 3 steps, and your cells are labeled. And also critically, we're able to label our cells before the experiment instead of at the endpoint. And so now with multiple colors, we can distinguish several different cell types in the same experiment. So now when we go to look at our, for example, our T cells mixed with our cancer cells, an example using flow cytometry, we can label one population of cells, in this case, some leukemia cells, violet. And at the end of our experiment, we can easily identify those. And using a viability dye called Cytox Deep Red, we can clearly resolve that at the end of 1 hour of mixing these 2 cell types together, the T cells have killed half of our cancer cells. So it's a very rapid and potent killing mechanism. And with this arrangement of 2 dyes in this case, we can clearly distinguish those cells that are alive and those that are dead. And this becomes a bit more complicated when we move to a type of therapy, such as a chimeric antigen receptor T cell, which is engineered to specifically recognize a marker, a cell surface antigen, on the surface of a cancer cell, and will kill only this cell. It will not kill cells that lack that receptor. And so now we can take this trick that we've described, label all 3 of these cell types with 3 different colors of tracking dyes, mix them together, in this case for 8 hours, and analyze with flow cytometry. And so what we'd expect to see is that our cells expressing CD19 are killed, and those lacking that antigen are not. And so the first result is that using flow cytometry, we can distinguish these 3 cell types from each other. And then we can just ask how dead are our CD19 positive cells compared to our CD19 negative cells. And so in this case, we've done all of the labeling and setup beforehand. At the end, we just take our cells, analyze them with flow cytometry, and get a result. There's no after the fact labeling or fixing or anything. So it's we're actually analyzing at the endpoint of this assay. And so we can take this method beyond flow cytometry and now into a solid tumor model, in this case, looking at a lung cancer spheroid, where we've made a ball of lung cancer cells and labeled our T cells red, allowed them to meet each other in a culture dish, in this case for 2 days. And we can see how our T cells will penetrate into this tumor model, and in green indicates those cells that have been killed by these T cells. And so this carries over well into kind of realistic models of solid tumors as well. Now, T cells are not the only immune cell that we're interested in, of course. Natural killer cells have some similarities to T cells in terms of their mechanism of killing, but their mechanisms of recognizing cancer cells are quite different. One mechanism of recognition involves an antibody binding to a cancer cell and a natural killer cell binding to that and then allowing a cell to kill. Although these NK cells are able to kill without any antibody present, just on their own, by recognizing specific markers on a cell surface that indicate this is a cancer cell. And so using a two-color kit developed by Scott Clark called the Live Dead Cell Imaging Kit, we can label some breast cancer cells with a combination of two dyes. Those cells that are green are live breast cancer cells, and they will turn red, and sometimes we see both colors present during that transition. They will turn red as they're killed by these blue natural killer cells. And so we're able to look in fluorescence microscopy at how NK cells can kill as well. We're constantly trying to make our methods more realistic and to better represent not just what happens in a cell culture dish, but what actually happens inside the bodies of our patients. And as I described previously, therapeutic antibodies can bind to a marker on a cell surface. A natural killer cell will recognize this binding and then kill whatever cell this antibody is attached to. And so again, using our solid tumor model, we can label a breast cancer spheroid blue, where the nucleus of every cell is blue. And we can take our natural killer cells now and label them red, add them to this breast cancer model for 2 days. And what we see is some penetration of those immune cells into this solid tumor and the presence of some green dots indicating cells that have been killed by those immune cells. However, if we now add a therapeutic antibody to the mix, in this case, Herceptin, a common breast cancer drug, we see it really looks like somebody has stepped on this spheroid. The killing is really quite dramatic, as is the penetration of those immune cells into the spheroid. But meanwhile, at the end, we can distinguish the two cell types from each other and report on which cell has been killed and which cell is doing the killing. I'll show you briefly how we can create an even more realistic model of immune cell killing, and that's by conducting the assay actually in the whole blood of a human or a human patient. I won't go into the specifics of how we do this today due to time, but there's a reference at the bottom for how to actually do this. But essentially, using an instrument called the Attune NXT flow cytometer, we can distinguish white blood cells in whole human blood from this massive amount of red blood cells and only acquire data on the white blood cells. And so we can actually label some leukemia cells, again, using our same trick, spike them into human whole blood with a therapeutic antibody called rituximab. And then using a viability dye, we will dilute this blood up to a very large volume and then run it, analyze it at a very high speed on our Attune NXT flow cytometer. And so again, just briefly, we can identify the nucleated cells, of which the native white blood cells are a fraction of those, but the cancer cells that we spiked into that blood are a substantial fraction. We can then ask, after 2 hours of sitting in healthy human blood, have these cancer cells been killed? And in fact, we can identify them and report, again, that more than half of them have been killed after this incubation. And so is that killing due to the therapeutic antibody that we added? And yes, if we leave out that antibody, we see that no killing happens. So it's entirely dependent on the presence of that therapeutic antibody. And so the thinking is that maybe this will permit the characterization of individual patients' blood for how they might respond to a given drug. But that can be studied in a test tube instead of the actual patient. So one final example looks at another type of immune cell. And this is a cell of the innate immune system, macrophage, which performs a unique function in that after it recognizes a cancer cell or a cell infected with a virus or bacteria, it will either take a bite out of that cell or actually eat the entire cell and ingest that entire cell into the macrophage. So besides my kind of crude cartoon depiction of a macrophage, it really doesn't do it justice. These are quite beautiful cells. If we label these as we see here in red with our cell trace violet, and then with a new dye called Cell Mask Deep Red actin tracking stain, developed by the chemist Zhangte Yang, and then developed in biology by Augie and Bosker, we can see the protrusions from this macrophage as they're exploring their environment, reaching out and sensing and looking for something that they should eat. And so if those macrophages identify a cell to which an antibody is bound, such as this one, they will then engulf that cancer cell. And so we developed a new method whereby we can label our macrophages with a tracking dye to keep track of them. And then as Dan introduced during the session last month, a pH-sensitive dyes called Frodo Green or Frodo Red, in this case, the AM ester of these dyes, so that the entire cancer cell will be labeled, the cytosol of that cell will be labeled with a pH-sensitive dye. So then when this cancer cell is eaten by the macrophage, it will become very bright in response to the low pH in macrophage organelles trying to degrade that cancer cell. So essentially, we label these two cell types and mix them together, and then we can ask, under a microscope, for example, using cell trace far red labeled macrophages, we can very easily identify individual macrophages because they are red. We can then take leukemia cells, in this case, and label them with our Frodo Green AM ester. And we see them as faint green spots in this assay, except for a few cases where a macrophage has eaten them. But if we now add a therapeutic antibody, rituximab, this encourages those macrophages to eat those cancer cells. And so we see that every red macrophage has eaten one or several of those leukemia cells. And in this case, this is only 4 hours later. So a very active process as well. And we can actually quantify this. So we can ask how green is every macrophage. And we see that when that therapeutic antibody is added, every macrophage on average becomes a lot more green, indicating that it has eaten a lot of other cells. So we went on to publish this method and demonstrate how it's useful for screening those therapeutic antibodies that will encourage macrophages to actually eat and destroy cancer by eating it. So the entire method is down here in the reference. But what went into that was a great deal of collaboration between our chemists in Eugene and our biologists, and also some great external collaborations with scientists in both academia and biopharma to make all of the different methods I've described possible. So we've looked at a few methods. I'm happy to talk in more detail about those, or really a lot of other things we're looking at. They're fascinating, and we could talk all day, but I want to give Bruce, actually, I'm really interested to hear what Bruce has to say about meiosis. So I want to thank the Knight Institute for organizing the session, and thank everyone for joining. Now, Alexia, I think I'll pass back to you.
Thank you very much, Chris. This is a great overview of some of the tools that could be used in immunotherapy research. I think we have a couple of minutes to open it for some questions. I don't see anything in the chat window, but if anyone has a question for Chris, would like to unmute and ask it. We have time for 1 or 2 questions. Just also a reminder that we've reserved 15 minutes after the hour for follow-up discussions with the speakers.
I have a question. So, Chris, I was wondering, these cell cultures, this is in 2 dimensions or in 3 dimensions?
Yeah, so are the cell cultures in 3 dimensions or 2 dimensions? And I showed examples of both. So the natural killer cell example, those were cells growing in a monolayer on a plastic culture dish. But anything that was a circle, those were actually 3-dimensional spheroids. So we're able to actually very easily grow a cancer cell line to form balls of, about 5,000 cells each. And we can actually image those using confocal imaging and image a slice through the middle of that spheroid.
I have one quick question. Oh,
go ahead.
I have one quick question for you, Chris. So you've kind of overviewed some of the tools we've developed. Looking forward, what do you think would be next? What are some tools that would be really valuable in this space that we could kind of start thinking about? Maybe not simple, but
well, we can always dream about what's an ideal situation would be, and then we can bring it to our scientists and see if it's possible. I think we've been looking for quite a while at more specific indicators, not just live and dead or tracking or things like that, but actually to build sensors that indicate a specific pathway has been activated. For example, when the T cells kill the cancer cells, specific enzymes are involved in that process. And if we can build a sensor that responds and indicates the function of those, then we can actually report very specifically what event has occurred. So that, and I think also we're seeing a lot of effort towards providing as many answers as possible from each experiment. And that means often more colors or interesting ways of combining a lot of sensors into one experiment.
thank you.
Chris, can you speak a little bit to how people typically quantitate the data, especially from a screening perspective? And have people begun to extend this to, the CAR T field, which is, I guess, very adjacent to most of what you showed?
Yeah, so as far as quantitation, it depends on the model. I showed some blood cancer experiments where often flow cytometry is used, where often the output teams are looking for is, with a given treatment, what % of my cells were killed in this case. And so this can be very low throughput with one well or one tube of an experiment, or very often, as I showed with the phagocytosis, these are very often expanded to 96 samples at a time, where, for example, in cell therapy, they're looking at, there was a study published last month by a friend of mine looking at the effect of the length of the linker in a chimeric antigen receptor. And it turns out that has a very large effect, but you need to be able to screen very large numbers of reactions and cell types to understand the result there. And definitely most of the first 2/3, actually entirely what I described relates to CAR T also, because we're now seeing chimeric antigen receptor natural killer cells, and we're working with a group developing chimeric antigen receptor macrophages. So you can actually reprogram your macrophages to target and eat whatever you want them to eat, usually a cancer cell. So yeah, thank you, David and Alexia, for the questions.
Thank you, Chris. And I think with that, we will switch over to presentation from Bruce. Just to introduce Bruce to our Thermo Fisher and U of O audience. Bruce Bowerman is a professor and the head of the biology department at U of O. He received his undergraduate degree in biochemistry from Kansas State University. And after 2 years at Stanford University School of Medicine, Bruce withdrew and decided to pursue a PhD on the integration of retroviral DNA into the host genome. And that was at the University of California, San Francisco. After receiving his PhD, he worked as a postdoctoral fellow with James Priss at the Fred Hutchinson Cancer Research Center, where he began to work on molecular genetic analysis of early embryogenesis in the nematode C. elegans. So with that, I'll hand it over to you, Bruce.
Okay, thank you.
Thank you.
So I can just share my, okay, all right. Okay. All right, well, thank you for inviting me to speak here. And as Chris mentioned, I'm going to talk about oocyte meiotic cell division, and our work is funded by the NIH because of its relevance to cancer biology. We study, cell division at a very basic level, trying to understand the proteins and pathways that execute different steps. And the idea is that incrementally that can lead to a better understanding of the process and help identify targets that can be used therapeutically. But we very much focus on the basic science end of things with all of our funding from the National Institute of General Medical Sciences. And so this, in fact, is a C. elegans worm, whole mount worm, showing the 3 most mature ovaries in the gonad. This most mature oocyte is about to ovulate through this thing called the spermatheca, where sperm reside and where fertilization takes place. And then these are fertilized embryos undergoing mitotic cell divisions in the uterus. So the earliest steps in oocyte meiotic cell division can only be imaged inside of whole mount worms. We're not able to isolate the oocytes in vitro, so we have to use whole mount worms for live cell imaging. And much of what my lab does is based on using live cell imaging to identify requirements for different genes during cell biological processes. And so in this example, we do live imaging by taking advantage of fluorescent protein fusions to various cellular proteins. So, for example, we can see the chromosomes here thanks to an M-cherry fusion to a histone that packages the DNA of the chromosomes. And so you see them in red. And then the microtubules we can see by virtue of a green fluorescent protein fused to tubulin, the subunits that make up the cytoskeletal polymers called microtubules that move chromosomes during cell division. And so this is the ovulation of an oocyte into the spermathecum. That's the oocyte meiotic spindle forming. And you'll see eventually it extrudes half of the chromosomes into a small polar body. And then it begins to undergo the second meiotic cell division. Oocyte meiosis consists of 1 round of DNA replication followed by 2 rounds of cell division. To reduce the genome from a diploid to a haploid content. So instead of having 2 copies of every chromosome, 1 from each parent, the oocyte is left with 1 copy of each chromosome, such that when a haploid sperm fertilizes it, diploidy is restored. I want to kind of give a little plug for our UO imaging facility. We've been working closely with Adam Fries, the director of the facility, both in using the microscopes available there to everyone here, and also his expertise in computational analysis of imaging data. And it's really been a productive interaction for my lab interacting with Adam. So I think, this has only been around here for a couple of years now, and it is a huge improvement to the core facilities and the infrastructure available here for cell biology research. And so as Chris alluded to, the cell divisions that occur in a body are mitotic cell divisions. They are equational divisions that maintain diploid, in contrast to the reductional divisions of meiosis that reduce a diploid cell to a haploid egg or sperm. And they also have very different spindle structures made up of these microtubule polymers I refer to. In mitosis, there are two microtubule organizing centers called centrosomes that define the poles of a spindle and both nucleate microtubules and provide a bipolar organization to the spindle. Centrosomes, just like DNA, are duplicated once every cell cycle, and so there are always two of them present. And when they enter into mitosis, they set up a bipolar spindle that can pull chromosomes apart. During oocyte meiotic cell divisions, you can see there is a dramatically different spindle. It's very small. They're, in fact, by this late stage in what's called anaphase of chromosome separation, there's almost no detectable pole microtubules, unlike in mitosis, and almost all of the microtubules here in blue are in between the separating chromosomes. So we're interested in how, and they look like this because oocytes, in fact, lack centrosomes. They're one of the few cell types that divide without centrosomes, and how you can make a bipolar spindle and nucleate the microtubules required for cell division without centrosomes is a very poorly understood process. So that's one of the things we're interested in. But today I want to talk about the sort of the final step of oocyte meiotic cell division, which is polar body extrusion, which refers to the extrusion of half of the chromosome shown here into a very tiny polar body. You want to preserve the large size of the egg so the embryo that ensues has all those nutrients. It's a highly asymmetric cell division that gets rid of extra chromosomes while losing as little egg cytoplasm as possible. And so that process involves the pinching in, ingression and pinching in of the membrane to kick out those chromosomes into a small membrane brown polar body. And in this movie, we're looking at a GFP fusion to the non-muscle myosin, which is a motor protein that interacts with actin microfilaments to provide the forces that cause the membrane to ingress and pinch off. And as you can see, this myosin is active not only at the site of polar body extrusion, but in fact, throughout the oocyte cortex, there's an active network of contractile actin myosin throughout the cortex. And as I'll tell you, we think that this network, in fact, is closely related to the assembly and function of the contractile ring that pinches off the polar body. So meiotic cytokinesis is in fact very distinct from mitotic cytokinesis, and mitotic cytokinesis is much better understood. During mitosis, it's known that the microtubules from the spindle poles, both from what's called the central spindle and the astromicrotubules, provide cues that organize the assembly and ingression of the actin myosin contractile ring that pinches the cell in two. Whereas during polar body extrusion, actually the ring forms distal to the entire spindle instead of midway between the poles, and then has to ingress halfway before pinching off to form the polar body. So the spatial dynamics of how myosin is regulated here are completely different. And while we know a lot about cytokinesis, in fact, we know very little about polar body extrusion and the process of meiotic cytokinesis. And so we got into studying this process because of a graduate student in my lab who recently joined Karen Oga's lab at UC San Diego as a postdoc, wanted to study a protein called CLS2, which is a member of the class family of proteins, which are proteins that stabilize microtubules by virtue of having what are called TOG domains that bind the tubulin subunits that form the microtubules. So they increase the local concentration of tubulin around microtubules to promote microtubule assembly and stability. And Elisa got interested in this protein because it was clear from the literature that it had an important role in oocyte meiotic spindle assembly, but that role had not been at all well characterized in the studies that referred to it. So she used CRISPR gene editing technology to generate premature stop codons that truncate this protein and basically generate null alleles that we can use to study its function during oocyte meiosis. And when we introduced that those mutations into transgenic strains expressing fluorescent markers, here we have a very nice pole marker called ASPEN1 that shows the two poles of the spindle, and then they kind of disappear as the chromosomes segregate. But you can see that in a CLS2 mutant, you never really get an organization of the ASPEN1 foci into a bipolar spindle. And in fact, the chromosomes form a single clump and cell division completely fails. So it's clear that this protein has important requirements during spindle assembly. We can also look at this in another strain with the microtubules labeled with GFP. And you can see again that it's a very disorganized spindle and never adopts the bipolar structure of a normal spindle, and it completely fails to segregate chromosomes. And in fact, the level of microtubules is also greatly reduced. The ratio of the intensity of the spindle microtubules to the cytoplasm is significantly lower than the ratio in the control. And this just shows that there's a loss of the microtubule signal. And that fits with class 2 being known biochemically to stabilize microtubules. They become less stable in this mutant, which fits with its biochemical activity. But what really got my attention about this mutant was that it completely fails to extrude any chromosomes into a polar body. And in fact, that was very penetrant defect for this mutant. It rarely succeeds in extruding any chromosomes into a polar body. It's a very penetrant defect. And that was surprising because there are anecdotal references throughout the literature to polar body extrusion being successful in many other spindle assembly defective mutants. But in fact, polar body extrusion has never been directly examined in severe spindle assembly defective mutants. So this kind of got our attention, and we decided to look at this process more closely using live cell imaging, which basically hasn't been done before. So we did a comparative study looking at polar body extrusion in 3 different mutants that have severe oocyte meiotic spindle assembly defective genes. The CLS2 mutants that I've showed you are defective, a kinesin mutant that makes a very clearly monopolar spindle, and then the most severely spindle assembled defective mutant we know of, a microtubule severing protein called MY1. When it's inactivated, you get no spindle poles, no organization of chromosomes, and basically almost nothing happens during oocyte meiotic division in a MY1 mutant. So we wanted to see, is this defect in polar body extrusion unique to CLS2, and what happens in other mutants with severe defects? So first, we just scored the success of pinching out any chromosomes that we didn't count, whether it was half of them, into a polar body that persisted at least part way into the following round of meiosis 2. And as we knew for CLS2, very few of the oocytes successfully do that, less than 20%. Whereas in a CLIP18 monopolar mutant, in fact, most of the time they do extrude chromosomes into a polar body, even though there's no bipolar spindle. And in fact, they often kick all of the chromosomes out into the first polar body, leaving an empty egg without any oocyte chromosomes. But in fact, CLS2 is not unique. We found that MY1 also fails to extrude chromosomes much of the time, even though in the literature it's been reported to have characteristically large polar bodies, which we now think result from an abnormal meiosis II cytokinesis, but we haven't characterized that in detail. So it was something of a surprise to find that in fact meiosis MY1 mutants also have a fairly penetrant polar body extrusion defect. So we looked at these defects using, again, live cell imaging now with a membrane marker that's fluorescent. And it's not easy to see these things. They're pretty small spindles, and we're kind of at the limits of spinning disk confocal microscopy resolution here. But you can see the membranes ingressing around the chromosomes that are being extruded and pinching off to produce a nice small polar body. Depending on the angle of the oocyte and the worm, sometimes it's harder to see, but this is a good example of what we can see. And it's almost always successful. Whereas when you look in CLS2 mutants, I don't want to dwell on this, but basically everything looks really weird. It's very defective. It's abnormal looking furrows all over the place. Sometimes we do see a little bit of chromosomes pinched out, but usually it's completely defective with highly abnormal furrowing of the membrane during the division process. And in fact, this is very different from both CLIP18 and MY1 mutants. CLIP18 mutants look pretty much just like wild type. They have nice furrows that ingress and pinch off the polar body, consistent with the usual success. In contrast, MY1 mutants actually, even though they're defective in polar body extrusion, the early stages look very normal with nice ingression, although it's a wider diameter, and substantial pinching in, here making a big polar body, but eventually this fails and releases the chromosomes into the oocyte with only rare successes. And so even though MY1 has a penetrant defect, it's a very different and later defect from what we see in CLS2 mutants. So then we also looked at these furrows with the non-muscle myosin to look at the process more directly. And this is the movie I showed you. We can use a computer to rotate these images in 3D and look down on the ring. So this is the same movie manipulated with the MARIS software. These are the chromosomes, and you'll see a ring form around them and pinch in and ingress. Okay, so that's what it looks like when you look down from above at the furrow. And so when we look in CLS2 mutants, you can see there's an attempt at furrowing, but it collapses back eventually and leaves the chromosomes in the oocyte. And when we look at it head on with the MARIS, you'll see a ring starts to form, but the myosin collapses into one big focus eventually and then disappears. So it has a very early defect, either in ring assembly or ring stability, that fits with the highly defective membrane ingressions we saw. And then again, in a CLIP18 mutant, basically things look really normal. You get good furrowing, good ring, good pinching off. And with MY1, it also makes a good ring that pinches nicely, but sometimes fails late. And so again, even though MY1 does fail, it's a very different late failure in the process. So a very different requirement. So the loss of CLS2, in fact, leads to a uniquely early and severe defect in contractile ring assembly or stability. And so we think it has a special role in the early stages of contractile ring assembly and ingression. And obviously this raises the question of what's it doing to promote the proper assembly and ingression of a ring? Well, if you look at where CLS2 is localized, it is at the spindle with these microtubules where you would expect it. It starts out all over the spindle and eventually is just in the central spindle microtubules. But it's also present in these patches throughout the oocyte cortex. And so it's conceivable that CLS2 could be doing something to influence polar body extrusion at the spindle, but it could be that it's doing something out here in the cortex away from the spindle that's influencing polar body extrusion. And in fact, it has another phenotype. Here you can see that during polar body extrusion of wild type, as I mentioned before, in fact, there's a contractile network throughout the cell cortex that causes small furrowing throughout the cortex. We call it global cortical furrowing. If you look at a CLS2 mutant, that global cortical furrowing is substantially exaggerated. It has a more active ingression throughout the cortex while failing to properly ingress around the chromosomes during cell division. And so maybe this phenotype throughout the cortex is related to the defect in polar body extrusion. This just shows we can take, we call these temporal overlays, where we take the membrane marker at each time point of a movie and then overlay them to show the ingression throughout the entire process of meiosis I cell division. And you can see that CLS2 is unique in having this prominent hyperactive furrowing throughout the cortex. And that may be relevant to the process of polar body extrusion. In fact, one model for polar body extrusion is that this global cortical furrowing generates a hydrostatic pressure, and the membrane within the contractile ring becomes depleted of actin and myosin and might be weakened. And this hydrostatic pressure might cause the membrane to pop out inside that ring. And if the pole of the spindle is tethered to that membrane, then it gets pulled out with the bubble of the membrane. And so that's one model for how polar body extrusion might happen, that it's not just ring assembly and ingression and pinching, but a coordinated effort of the actin-myosin cortex that is promoting polar body extrusion, perhaps through this hydrostatic pressure thing. And to save time, I'm going to skip this. There's a mutant that has upregulated myosin activity that sometimes extrudes the whole polar body. So the idea is that it has more hydrostatic pressure, makes too big of a bubble, and drags the entire spindle and all the chromosomes out with it. So we looked at actin-myosin in our CLS2 mutants to see if it's acting like that other mutant. But in fact, actin-myosin looks completely normal in a CLS2 mutant. So it doesn't seem that upregulation of myosin is the problem, unlike in this other mutant that's called CSNK1. So we proposed an alternative hypothesis that CLS2 patches throughout the cortex might promote cortical stiffness by stabilizing cortical microtubules. That's what this thing is. It's a microtubule stabilizer. And maybe stabilizing cortical microtubules makes the cortex more stiff and resistant to ingression in response to the actin-myosin contractile forces. So you get rid of CLS2, the cortex becomes less stiff, and the same amount of myosin activity can cause greater furrowing, is our hypothesis for how CLS2 might be affecting this cortical furrowing business. And so Alyssa Kweokwe is a rotation student in the fall who addressed this, and she tested this idea by asking, can increased levels of cortical microtubules suppress the extra furrowing in CLS2 mutants? In other words, if you add microtubules and make the cortex more stiff, does that then suppress and prevent that extra furrowing? And we know of mutants, I won't tell you what they are, they're more microtubule regulators called CLIP7 and ZYG9, that if you knock them down, have an increase in cortical microtubules relative to wild type. So we put these into our CLS2 mutant background, and they still have an increase in the level of cortical microtubules. So in fact, these extra microtubules don't depend on CLS2 to be present. And so we can now see if they can suppress the furrowing in a CLS2 mutant. Again, these are our temporal overlays in wild type, the extra furrowing and CLS2 mutants. And with preliminary numbers, Alyssa is going to come back to the lab this summer and finish this up. In fact, we do see that CLIP7 knockdown and ZYG9 knockdown in a CLS2 mutant does appear to suppress the extra furrowing. And in fact, I don't really have time, very recent data from a new research associate, S.K. Sumiyoshi in my lab, is finding that in CLS2 mutants, we see a decrease in bright foci of microtubules throughout the cortex. So this may fit with our model that CLS2 reduces the levels of, not maybe the levels, but the organization of cortical microtubules. And so this supports our hypothesis that CLS2 patches promote cortical stiffness by stabilizing cortical microtubules. And so the, we still don't know exactly what's going on here, but we think there is a relationship between cortical cytoskeletal dynamics and contractile ring ingression and polar body extrusion. And we want to continue to study the regulation of cortical cytoskeletal dynamics, both myosin and actin and the microtubule cytoskeleton. And we think a balance of stiffness and contractility are probably important for this relationship between the cortical cytoskeleton and polar body extrusion to function effectively. So I want to thank Elisa, former PhD student, and Alyssa, a current PhD student, and Esuke Sumiyoshi, who is not in this picture, but this is the guy who got him to my lab, for their contributions to this work. And I'll stop there and take any questions.
Thank you very much, Bruce. This is a very nice overview. There's a question in the chat box from Sean Burrows. He's asking, is the cortical stiffness hypothesis an alternative, or is it complementary to the hydrostatic pressure model?
Yeah, that's a good question. I don't think they are mutually exclusive. I think they could both be, related. The stiffness and the contractility balance out, maybe to give the right amount of hydrostatic pressure is one way to look at it. Or they could be operating completely in parallel. The one thing I didn't go into, just to try and keep this within the time limits, was that, in fact, I'm going to reshare my screen. Oh, this has happened to me before. Never mind about the sharing. That model where I showed the spindle being pushed out into a bubble, one problem with that model is that actually that oocyte is ex utero and it's in hyperosmotic medium that causes the oocyte to collapse away from the eggshell to where the spindle can pop out. In fact, in a worm and in a normal embryo with physiological salt conditions, the membrane is right up against the eggshell and you don't pop out a bubble, you make a ring that ingresses. And so I think there's a relationship between the cortical cytoskeleton and the furrowing, but I don't think I don't think this hydrostatic pressure model completely makes sense in a completely normal context. But actually, that is what mouse oocytes do. So that's another reason to favor that model. We're still trying to wrap our heads around these models, and they're not mutually exclusive.
Thank you.
Thank you. Oh, thanks.
I have a quick question around the time-lapse movies. What are the time frame?
Yeah, I didn't go into, we could give a seminar just on making these movies and doing the image analysis. In fact, I forgot to mention our collaboration where we're working with the imaging facility with Adam Fries to automate and our computational analysis of these images to score the number and depth of cleavage furrows and to score the number and intensity of the microtubule foci that we're seeing throughout the cortex to provide good quantitation for these phenotypes. And so to make those movies, it's always a balance between the bleaching of the fluorescent proteins and the time resolution and intensity of the images. And by playing around with laser intensity and the various conditions, basically we typically take a stack of 10 or 20 focal planes in a Z stack, and we can do that every 10 seconds is kind of our standard imaging. If we do less of a Z stack, we can do more frequent time points. We can play around with that, but most of the movies I showed you are from that kind of format. Cell division happens very rapidly in these oocytes and early embryos, which is one virtue for using C. elegans to study this stuff. That cell division would take 24 hours in a mammalian cell culture. It takes 20 minutes in a C. elegans embryo. So
I see. Okay.
yeah, and hopefully, some of this stuff leads to people like Chris being able to make inhibitors that stop cell division, but it's a very incremental business doing basic science.
True, but very informative.
Hey, Bruce, I had a question. You showed a lot of, really intriguing models, but just practically, it seems like a lot of these should be lethal mutants. How do you keep these going long enough to study them and
Sure, yeah. In fact, this is a lethal mutant. And so we carry that mutation in a strain that is heterozygous for the mutation. And then we can isolate homozygous mutant progeny. And we are pretty lucky that for this gene, those homozygous mutant larvae survive to adulthood and are fertile and make embryos that have defects and then die. In fact, most cell division genes, if you completely knock them out like we did with CLS2, the homozygous mutant animal would either die sometime during larval development or become a sterile adult due to defects in cell division during larval development. And it just so happens that this gene isn't required for all the cell divisions leading up to a fertile adult, but only acts in the early embryo. So it's kind of unusual. For other genes, we can use RNA interference and introduce double-stranded RNA into these adult worms that are otherwise wild type, and that knocks down gene function in an adult fertile worm. We can also tag genes with what's called a degron tag, and then you can treat them with a plant chemical that converts that degron tag into an E3 ligase basically and leads to the degradation of the protein. So there are conditional ways to reduce gene function in fertile adults. And in fact, the Zyg9 and CLIP7 work was all done with RNAi because you can't work with a complete loss of functional allele. You wouldn't get a fertile adult. So we do various genetic tricks. We can carry lethal mutations as heterozygotes and then isolate the homozygous progeny, or we can use these more modern methods to reduce gene function with RNA interference and degron tagging. We also used to isolate lots of temperature sensitive alleles of these genes. And then we can use temperature upshifts to inactivate proteins at specific times. So there are various ways of conditionally inactivating genes that we take advantage of.
Okay, thank you.
I don't see any more questions in the chat. Is there anything else folks on the call would like to ask or share with Bruce?
All right. Well, thanks very much for sponsoring these talks. And
I have one question.
Yes.
Last one probably here. When you correlate these pathways with what happens in cancer cells or mammalian pathways, do you see any similarities or in the cell division mechanisms and actin dynamics?
Is that a question for me?
Yeah.
I didn't understand. What was the...
No, you showed a lot of meiotic division and also the stiffness actin dynamics in the. And what I'm asking is there any correlation between these pathways with what happens in the cancer cells or?
Yeah. Well, to be honest, I don't know. That's certainly true that the cortical actin myosin cytoskeleton has important roles in cancer, with cell migration being the most obvious one, and which is relevant to metastasis. And so I do think that by studying just the fundamental regulation of cortical cytoskeleton in a model system like this, does end up leading to insights in the regulation of cytoskeletal functions in cancer cells that prove relevant. But I'm not aware of any direct connection between the CLS2 gene we're studying and cancer biology. But I wouldn't be surprised if there are at least some association type studies that implicate them in cancer. Many cell division genes do get implicated in cancer eventually.
Okay, thanks.
Hey, Bruce, Bhaskar, who asked the previous question, made me realize over the last couple of years, he and others on our team have developed small molecule actin and tubulin trackers. They're deep red versions, and just in the spirit of collaboration, it would be interesting to send those your way.
I was struck by that deep red looked really pretty with the macrophage, I think it was, and it might be that we could use some of those. And those are small molecules. One problem we have is that these embryos produce a chitinous eggshell pretty quickly that is impermeable to a lot of small molecules. But there are ways around that, and people have used like LifeAct and some other commercially available things to look at actin microfilaments. And it would be interesting to play around with the deep red, actually. I'd be curious to look at that. In fact, we struggle to look at microfilaments somewhat. The various fluorescent protein fusions to microfilament proteins often cause functional problems that have made it difficult for us to use them. So that actually might be very useful for us.
Well, maybe we'll connect right after this, and there are various colors of both actin and tubulin. We can send those your way. It'd be really interesting to see their behavior, both with the eggshell challenge and
and if they got through the eggshell, then it would be golden. But even if it can't, there are ways. One can poke holes with a laser beam, or you can use eggshell defective mutants, but there are challenges to both of those approaches. But and one could maybe even like grow the worms on them, and it would get in through the gonad and stuff. So there are ways to play around. I'll send you an email, and we can pursue that. I think that's worth doing.
Great. There may even be like a bike courier delivery associated with that.
Sounds great.
Thank you very much, Chris and Bruce, for very nice talks. And it was great to see some of the research going on at U of O. Some of us who are graduated from there are familiar. But for those of us who aren't, it's great to connect with all of you and learn what you're doing. So I think we can wrap up if there are no other questions. Tony, are we good to?
I think that's great. Yeah, thank you everyone for taking part. These presentations were fantastic. I'll also make sure Chris and Bruce, you have each other's emails.